Okay, good morning, everyone. Welcome back to the SAEON Seminar series. We were just looking at the history, and this is about the 25th talk we've had already in the series, which is great. So this morning, it's the turn of SAEON and Ndlovu node again. And our scientist, Dave Thompson, is giving a talk this morning on a project that's actually been running, small project, but one that's been going a long time at the node. For those of you who don't know Dave, he joined SAEON all the way back in 2008, actually as a postdoc first at the node, and then the following year became our scientist. He's been there ever since. So, as we all have. So let me hand over to Dave, who's going to talk to us this morning about an endangered species, the pepperbark tree, which also has important social value. Thanks, so Dave. Over to you.
Just keep everyone busy for 1 minute.
Oh, okay. You're not going to start just yet. There were protesters blocking the roads into Palabora this morning, so hopefully the office is not being attacked.
Yeah, I just had to plug my laptop in. It just suddenly shot up a message saying the battery was low.
Okay.
Yeah.
All right, over to you. Thanks.
Great. Thanks, Tony. Yeah, so my title is up there. It's like Tony said, it's work that I've been doing or been involved with since 2013. And it's really looking at the natural history of the pepperbark in Kruger. And some of the sort of the disturbing findings that we found regarding the possible persistence of the species. So I just need to start off by just mentioning some co-workers and the funders. A lot of the work's been done by a group of people at WITS, two amazing MSc students, Kaylee and Semester, and then their supervisors, Glynis Cron, Kelsey Gannon, and Ed Witkowski. And also two partners from Stellenbosch, although Yaku LaRue has now moved to Australia, but Yaku and Megan sort of drove the genetics component early on. Of course, I need to acknowledge Sandparks for the support, and then the SAMBI FPIP program for funding, as well as the Botanical Society and the NRF. So those of you who know me as someone who normally has my head down in the herbaceous layer of grasslands or savannas. I have a soft spot in particular for the flowering plants. And usually what I'm looking at is the effect of fire or drought, some kind of disturbance or grazing on how that structures the herbaceous communities in those systems. But we're not going to be talking about any of that today. And we're also not going to be talking about cheetah today. But the cheetah does serve as a very nice model for some of the concepts that we're going to be touching on today. The cheetah sort of came into the spotlight a couple of decades ago as a species sort of, very genetically compromised. It went through a bottleneck event and so on. So we're going to just sort of go through that process of how a species ends up in that position where it's facing extinction. And the first sort of step is you get fragmentation and decreased population density, which results in reproductive isolation. You get a reduction of gene transfer, which results in inbreeding depression and something called the alley effect. And the critical point there is that you have these populations then which end up with reduced genetic diversity. And the big implication there is that those populations have decreased adaptability, decreased resilience, and decreased flexibility in terms of environmental change. And so what you end up with is some kind of effective population, which is a lot smaller than the actual population. And if you then encounter a population bottleneck event, you tend to end up with a system where the genetic diversity is so low that you basically on a trajectory to extinction. And then I've just indicated here there are corrective interventions that you can apply sort of earlier on in this process. Later on in the process, it's a little more difficult to intervene. So these are some of the concepts we're going to be thinking about today. And just two that we want, also I want to touch on in a bit more detail, that might not be familiar to a lot of people, is the alley effect. And really, this is just the relationship between, sort of a mathematical relationship between the size of a population and the fitness of the individuals in that population. So the reproductive capacity of individuals in that population. And typically at some kind of intermediate population size, the individuals in that population have achieved their maximum reproductive potential. But in populations, sort of if you look at the red line here, populations that have sort of a strong alley effect at low population densities, the fitness of the individuals is actually so compromised that the population has a negative growth rate. So in effect, the population is kind of on the sort of self-sustaining trajectory where the population is getting smaller and smaller. The second point just to touch on is the concept of the genetic or the population bottleneck. And this is some kind of collapse due to a stressor. It can be environmental stress, it can be an anthropogenic stress on the population. But essentially, it's a massive collapse in population size, which really gets to a point where either you need to, the population can recover or it's on the trajectory towards extinction. And this is where genetic diversity really plays sort of a really critical role, because it's that sort of diverse gene pool that either gives the population the capacity to resist the bottleneck event or to recover from the bottleneck event. Okay, so just backtracking a little bit to some basic biology, just to sort of get some concepts clear in our mind. So most people, or certainly most vegetation ecologists, focus here on the adult stage of the population. If you're sort of generous, you might include some sort of seedling work. And so what we typically do is we run out into the field with our height poles and our tape measures and our quadrats. And we take a bunch of measurements and we make some sort of conclusions about the health and the status of the population. And sort of an example of that is sort of a size class distribution, where healthy populations tend to have lots of smaller individuals and less mature reproductive individuals. Okay, but a lot of this sort of reproductive cycle tends to go unstudied and sort of uninvestigated in terms of population health. And it's really, it's not just at these stages here where population status can be determined. It can be anywhere within the cycle. And so I just want to spend a minute here just looking at this sort of magical stage between where you go from being a plant to a plant with fruit, because I mean a lot is happening between those 2 points. And from, in terms of a sexually reproducing individual, the most important part is the formation of the gametes, which introduces genetic diversity, and then the pollination event, which is a random recombination of gametes. So again, introducing genetic diversity. And so this sort of stage of sexual reproduction is critical because it introduces the adaptation capacity that a population needs to sort of survive in a changing environment. It does have some downsides. Sexual reproduction is expensive and wasteful, and it's often resource limited. And I've just sort of highlighted here, this stage here is critical, but there are other stages in the process: dispersal, fertilization, germination, recruitment, maturation. At any of these stages, the population status or fate can be determined. And then we can't sort of talk about sexual reproduction if we don't just mention that plants, or at least most plants, have available to them the option of asexual reproduction, where sort of the whole sort of bottom half of the cycle here is bypassed, and the adult individuals produce new offspring through the production of genetically identical clones. And this is useful, it's cheap, but it does have the disadvantage of reducing genetic diversity and adaptation capacity at the population level. And so then we have this system in plants where the reproductive function that the plant is expressing is a trade-off between the asexual and the sexual function. And for many plants, it can be either or, either of those, or it can be both of those. And the critical point is that the sexual function, or reproductive function rather, is not fixed, it's dynamic over time. It can change over the lifespan of an individual. And then it also changes over, much longer evolutionary timescales. And then finally, we get to the pepper bark. It's Warburgia salutaris. It's a medium to small sized tree. It's got these sort of small yellow green flowers and these fruit which look a lot like olives. It occurs in montane forest and sandfelt forest, as well as sort of high altitude rocky ridges in eastern and sort of southeastern South Africa. Yeah, so now that we've introduced the concepts and species, let's have a look a little bit more detail. So in terms of the context of the species in Africa, it's critically endangered. And one of the main reasons for that is sort of high levels of exploitation. It's a very commonly used medicinal plant. Warburgia salutaris here in the purple used to be distributed right up through Malawi. At the moment, it's not found in Malawi, Zambia, or Zimbabwe. It's locally extinct. And the current sort of distribution is through northeastern South Africa, through Eswatini, and in southern Mozambique. And in all those countries, it's got some kind of conservation status, either endangered, critically endangered, or vulnerable. Like I mentioned, it's subject to intense harvesting pressure for traditional health systems. It's mostly the bark that's harvested, which is not really sustainable. And so there's obviously conservation concerns in around the species due to the fragmented nature of the population, these sort of high levels of harvesting and the impact on population density, and then concerns around the genetic diversity and the capacity of the species to adapt. In South Africa, it's also forms part of our traditional health system. In recent decades, there's been a 50% decline in South Africa, and there's decreased distribution, decreased number of populations, less individuals, more fragmentation. And we're even finding that in terms of sort of health or traditional markets, the bark products are now being imported from Mozambique because it's increasingly hard to find in South Africa. It's primarily restricted to protected areas at this stage, including Kruger Park. I'm sitting up here in the extreme northeast. Mpumalanga and Limpopo are currently the strongholds, but there has been considerable local extirpation. If we have a look at the map here, the crosses are where it used to occur in sort of South Africa, and the dots are where it currently occurs at the moment. And a lot of these subpopulations that are existing have very few individuals, often, or in some cases, less than 20 individuals. And again, we come back to this point where there's concerns about the gene flow and the possible impacts on the persistence of the species. Just to point out here that as a species, it responds very nicely to damage, or it responds positively to damage in that it produces coppice growth, which results in individuals with lots of stems. And so if you're doing stem counts, you might get a sort of an artificial impression of the size of the population. So that's just something that we need to keep in mind as well. In terms of then the Kruger Park population, it's limited just to the high elevation areas in the extreme northwest of the park. It occurs only on south and southeast facing ridges. There are 13 subpopulations, and these range in size from 1 to estimate for several hundred individuals estimated. Something that's important is that the populations lie very close to the Kruger Park boundary. This is the boundary here. Some of them are within a couple of 100 meters of the boundary. This is a view from one of the populations looking out of the park. The populations are at most 10 kilometers apart. And despite the fact that they're occurring in the park, there's still ongoing bark poaching. And there's just a view, sort of a picture, sort of in-field picture. This is Warburgia here. There's one larger individual and then several smaller individuals at the bottom here, just so you get an idea of what it looks like. And then in terms of the Kruger sort of context, Sandparks is running a very active Warburgia program, and this is underpinned by 3 sort of pillars, and the first, so I've grouped them into sort of multiply, protect, and monitor along the bottom here. And so the multiply pillar is really to promote the sustainable use of the species, and really that focuses on providing a material sort of outside the park for use by communities and so on. And it's been a very successful program. Initially supported by micro or vegetative propagation using cuttings and micropropagation, and then more recently by seed, and we'll talk about that a little bit later. The second pillar is to protect the species, and basically just to reduce illegal harvesting within the park. And there are armed rangers patrolling to look at the, monitor the populations. And then the last pillar is to monitor. And this is where SAEON really got involved in the program, and this goes back about a decade now. But really just to get a sense of what the population in Kruger is doing, how it's behaving, and how we can use that to guide some of the management interventions that the park needs to implement. And this monitoring involved stem counts, measures of height and stem diameters to get some of those sort of size class distribution maps, or sorry, size class distribution figures that we looked at earlier. And then also to look at some of the levels of poaching. And so what did those surveys in the park show? Well, the first finding was that the population seems to lack juveniles, and that's concerning because it indicates a lack of recruitment. And that's probably driven by the fact that there was some period in the past where recruitment from seed was favorable, but it's not favorable anymore. About 65% of stems in the park show evidence of debarking, which is a really high number. And then, like we said, it responds really well to disturbance. And so most of the individuals, or at least half the individuals in the park, have more than one stem. And this sort of speaks to some ongoing disturbance, which is likely the harvesting in the park. All right, so we get to a point then, is like, how did I get involved and what was I trying to achieve as part of this bigger project? Well, the first thing that was sort of concerning to me was this lack of juveniles. And so we really wanted to sort of get to understand what was driving this lack of recruitment. And then two sort of ad hoc sort of in-field observations really got my interest. And the first was that the reproductive output is really limited. Flowering is incredibly sporadic. You hardly ever see trees with flowers. When you see fruit, if you see them, they're very scarce. They're normally parasitized by marula fly larvae. And we didn't find any evidence, we didn't see any seedlings growing in the population. But then something that we did also notice was that where the roots are damaged, or where the roots are exposed above the surface, the species responds by producing root suckers. Okay, so it's basically able to reproduce vegetatively as well. And then looking into that further, we found that there has been some reports from KZN of the species producing clones. And so we got to this point where we have this kind of trade-off, this antagonism between sort of what looks like failing sexual reproduction and the capacity to reproduce asexually. And so then we had sort of three aims: was to then look at the prevalence or the, was the Kruger population clonal and how prevalent was that? Try and unpack the failed sexual recruitment in the population, and then of course look at some of the implications of that on what it means for the persistence of the population in Kruger and its capacity to adapt to change. So we're going to have a look first at the genetics and then at the reproductive component. So basically this is the same, the map of the subpopulations in Kruger. And we sampled 258 individuals across those populations. And when I talk about individual, I'm not talking about, say, multiple stems from one individual. I'm talking about stems that were at least 5 meters apart. Sorry, we did 258 individuals total. 227 came from the Kruger subpopulations. And then we included 31 individuals that we took from a variety of different nursery stock populations outside of the park. Okay, I'm not going to go, there's like 3 lines there about the methods we used for the genetics, but basically we developed some polymorphic microsatellite markers. analyzed the data using POP-R for measures of clonality, structure for genetic structure, and genodive for genetic diversity. The first thing to look at here is the measures of clonality, which is basically a test for linkage disequilibrium. And we had this significant deviation of the histogram here to the left of what we expected, which is here at the blue line. And so this deviation suggests that there's linked loci within the population. And that's indicative of prevalent clonal reproduction. If we look at how those clones are distributed in the population, so in Kruger, we sampled 227 stems, but we only turned up 113 genotypes, which means that half the stems in Kruger share their genotype with at least one other stem. So at least half the stems are clonal. And those clones ranged in size from having one stem to up to 23 stems within a single clone. Just a couple of examples here. We had one subpopulation, had 14 stems, and they were all of a single genotype. So the entire subpopulation is just one clone. At the other end of the extreme, we had a subpopulation of 31 individuals. Most of those were unique. So you have this range from some populations which have multiple genotypes to others which have a single genotype. The biggest Kruger population, we sampled about 100 stems. We only turned up 25 genotypes. So that means that 75% of the stems in that population share their genotype with one other stem. Okay, so 75% are clonal. And in that population, we found that 23 stems were from the same genotype. Okay, genetic diversity. This returns sort of a bunch of genetic diversity metrics, including Shannon's diversity. And so this took we had to take a step back here because it was sort of paradoxical what we found. We turned up high genetic diversity in Kruger, especially where there was many genotypes within the subpopulations. And this is maybe initially not what you would expect, but we sort of looking into it, we found that the levels we got was within the range of what is expected for clonal organisms, especially if the loss of sexual function is recent, or if there's a high accumulation of mutations within the genotypes. Okay, so we're sort of happy that although we didn't expect it, we did get these high levels of genetic diversity. Just to sort of expand on that, there's the population we spoke about that had just one clone. Of course, the diversity is low. In fact, there's no diversity. Some of the bigger Kruger populations where there were multiple genotypes, we did have these high levels of genetic diversity. We also had fairly high levels of genetic diversity in the nursery plants. And that's perhaps not unexpected either because these plants had been taken or originated from different or multiple localities. Sort of visualizing all that in one one picture, we look at this minimum spanning network of similarity. And so the distribution in space shows the genetic diversity between the different genotypes. Small dots, so some of them are genotyped with single stems, others are genotyped with multiple stems based on the size of the bubble. That's the purple dots. And then the turquoise dots are the nursery plants, and they are distinct for the most part from the Kruger population, mostly unique individuals. There are a couple of multiple genotypes there. And it's important here to notice that even though the nursery populations are similar in some cases to the wild population, they are never identical. All right, so let's have a look at this. What does this look like on the ground? So there's the largest of the Kruger populations that we sampled. We sampled 100 stems there. And so you've got the situation initially where you walk in and you think, okay, we've got 100 stems from 100 individuals. But if you look at the same picture where you've plotted it according to clone, you suddenly see a situation where you've got, instead of a linear population, you've now got clusters of clones spread across the landscape, where the shapes here with the same color are stems sharing genotypes with each other. So here with the green circles, all those stems belong to the same individual, whether they're all clones of the same individual. Okay, the second sort of aspect of the project or the program was to look at the reproductive ecology. Here we are focused on the 4 largest subpopulations in Kruger, and then also a subpopulation that was discovered after we did the genetic work, where some old fruit had actually been found previously. Flowering is in June, May and June. And fruit set is in the middle of summer, November, December. Okay, so what did we do? Well, in those those populations, we only turned up 42 flowering trees out of several hundred. And what we did is we used those flowering trees to conduct a series of pollination trials. And these, they're listed there, but basically tried to assess whether the trees can self-autonomously, which is where you move pollen within one flower, the same flower, from the anther to the stigma of the same flower. Diatonogamous selfing, where you move pollen from one flower to another on the same tree. And then outcrossing, where you move pollen from one individual to a different individual. And then we also had some options where we allowed natural pollination to occur, and we bagged the fruit, or the pollinated flowers, to see if we could protect them from that parasitism by the fruit flies. Okay, so what do we find? Autonomous selfing doesn't result in any fruit initiation, and other pollination trials also resulted in really low levels of fruit initiation. Okay, at most they're 4.5% about 2%, and then the natural pollination 3 to 16%. So this is now fruit initiation. So how many of those pollination flowers initiated fruit? Okay, but then if we take it a step further, if we look at the maturation of fruit, only 3%, or not even 3%, of natural pollination resulted in mature fruit, and that's 3% of the 3 to 16%. So these numbers of fruit production are incredibly low. And so what we've got then is a population sort of characterized with low fertilization coupled with high abortion, and this is of course concerning. Looking at the pollination syndrome, this had never been documented for the species before. There had been some sort of notes in various publications suggesting that because the flowers are small and green, it could be, pollinators could be solitary bees, small bees, or perhaps wasps, which tend to like those kind of flowers. Pollination observations turned up a variety of butterflies and moths, flies, beetles, wasps, and bees, and then also thrips. But based on the abundance, based on the way that the pollinators interact and contact the reproductive parts of the flower, and then based on pollen load counts on the proboscis of the different pollinators, it's very clear that the Lepidoptera, so the butterflies and the moths, are the primary pollinators of Warburgia. We also looked then at pollen viability. Okay, so just some tetrazoleum staining to look at which pollen grains are viable, and we found fairly high levels of viability, up to 93%, depending on which subpopulation the pollen was taken from. And then something else we looked at was the ability of the pollen to germinate, if we can use that word, and produce pollen tubes, which eventually take the gamete down to the ovule. And irrespective of the cross, so irrespective of where the pollen came from, once the pollen reached the stigma, about 32% of the pollen grains were able to initiate pollen tube growth, but only 7% of those tubes actually reached the ovules. Okay, and there are genetic reasons why that might be the case. Okay, this is, if you're familiar with the terms of self-incompatibility, this is one of the primary stages where self-incompatibility is kind of activated, where basically the genes of the pollen, the genes of the stigma are not compatible. Pollen production overall was quite low, but not necessarily so low that we'd be troubled by it. But what was concerning is that in some populations, we noticed that the anthers, although they matured, they never split and released the pollen. Okay, so the pollen was never then in circulation in the environment. And that failed anther dehiscence is actually a documented form of genetic male sterility. Okay, so we're getting a clue here that maybe not all is right with the genetics of the population. Okay, so we mentioned earlier fruit set is very low. We only found mature fruit in the largest population in Kruger. In 2019, only 2 stems of the same clone produced fruit. 2020, those same 2 stems produced fruit plus 1 stem on the second clone. Okay, of the few fruit that were found, 9% have this sort of damage to the immature ovule where some kind of insect with a sucking proboscis has penetrated the ovule wall. And then another 20% of the fruit are parasitized by the maroon fly at some point. Okay, so again, we come back to this point where you have this population where you have very few flowers, very few fertilization events taking place, and of those fertilization events, most of them are being aborted. Yeah, up to 91% of fruits which are initiated are aborted before they reach maturity. And so there's 2 hypotheses behind this. The one is that there's some kind of resource limitation. The other is that there are some kind of developmental lethals in play. So these are genetic factors, either activating self-acting self-incompatibility or activating post-zygotic failure. So after the zygote is formed, the abortion is driven by genetic incompatibility. Okay, so having said all that, we did manage over 2 years to find 22 fruit that matured. From those fruit, we extracted viable seed. Those were grown and included into some of the stock populations in the nursery. Okay, so we had very little evidence of full reproductive capacity in Warburgia. Okay, so if we measure reproductive capacity in terms of mature fruit being produced with viable seed, that was limited to just 3 stems of 2 clones. And the implication of that is that most of the genotypes in Kruger have lost their sexual reproductive capacity. Okay, and it doesn't have to be that way. And I include here a picture or pictures. This is an artificial, it's a sort of an orchard, if you want to call it that. These are Warburgia trees that were planted in the KZN Midlands about 30 years ago. The seed for this population was sourced from False Bay in northern KwaZulu-Natal. And this population produces prolific fruit, masses of fruit. And what's interesting is that it's out of range. It's not planted within its natural range. And the one sort of idea behind this is that it's much wetter and it's not as hot as Kruger. And so that might be contributing to the success of this population. And we spoke earlier about how the Kruger program was producing sort of thousands and thousands of plants to give to communities to relieve the pressure on harvesting. And it's the fruit from this population that are being used to underpin that propagation program. Okay, so clearly the species has the potential for reproductive output. Question is, why is it not achieving this potential in Kruger? And so there's sort of a couple of things we need to talk about here. Okay, so we have a population that has some reproductive potential in Kruger, does produce flowers, although in low numbers. There's abundant pollinators, the male function is not compromised. And if you can get a fruit through to maturity, it does produce viable seed. On the other hand, we have this population which is characterized by chronic lack of mature fruit. There are very low fertilization rates, very low rates of embryogenesis, and incredibly high levels of fruit abortion. And so we've got 2 hypotheses in play here. The first is the activation of the developmental lethals. And so you could then say that in Kruger, the population is primarily outcrossing. Okay, it relies on pollination between different individuals, or in this case, different clones. But because the flowering is so sporadic, and because the stems that are growing next to each other are typically stems from the same clone, the population is effectively then self-pollinating. Okay, and we showed that it's not capable of self-pollinating for genetic reasons. Okay, the other issue that comes into play here is that sort of evidence we found that the genotypes might be accumulating sterility traits, like the inability to release their pollen. The second point to think about is the resource limitation hypothesis. And this is where fertilization can take place, but for some reason, the plant allocates its resources away from fruit production towards vegetative growth or regeneration. And the implication here, or the thinking here, is that this would be characteristic of a population that is in disequilibrium with its habitat, with its environment. Okay, the current conditions are not suitable for the plants to provide resources towards sexual reproduction. Okay, so we have a population then which we could say has had fairly recent failure of its sexual reproduction or sexual function, and that in Kruger the plant is persisting mainly via clonal reproduction. And the unfortunate thing here is that this is sort of self-sustaining, and that the longer they persist as clones, the further the sexual reproductive capacity is lost. And so what is the fate then of the pepperbark in Kruger? If we go back to our sort of cheetah model, so we've shown that population is fragmented and the population densities are decreasing. Because of the population sizes, the allele effect is probably in play. We've then got evidence of reduced gene transfer with a limited reproductive function, the reliance on clonality. Although we didn't show that genetic diversity was low, we can say that the functional genetic diversity is low because the diversity is held within the genotypes and it's not in circulation in the population. And so we could conclude then that the effective population size is much lower than what we thought. And so the question then is the population viable? And the answer to that question is then, do we proceed past this sort of threshold point to a population bottleneck event in extinction rather. And so in order to determine whether we sort of go past the threshold is whether we know what kind of corrective interventions could we possibly implement here that are going to change the trajectory of where we're going with the Warburgia. So if we look at that, also coming back to that figure we looked at earlier, we have this population with lost reproductive capacity and a severely compromised adaptation capacity. And so I feel like the population is really sitting over here. and it could be some sort of bottleneck event in the future, either a climate change event, such as a temperature or rainfall threshold being exceeded, some kind of extreme event like a drought, could be continued harvesting pressure, disease, other anthropogenic effects. But it's not going to take very much to shift the population from here down to here. Okay, and then you've got the situation where do we proceed to local extinction in Kruger, in which case we sort of turn that to another cross on the distribution map, or will the population have the capacity to recover? And so some of those possible interventions that we could look at in terms of changing the trajectory here, and I've sort of grounded this in the three pillars of the SANParks Warburgia program, the first being to multiply. Okay, so there are stock plants outside the park. There's lots of them, and they are diverse in themselves, but they are distinct from Kruger. Okay, so the Kruger gene pool is not captured in ex situ populations, and so one of the priorities there is to really get those Kruger genotypes into ex situ collections. And this can be done in two ways. The first to be via seed, which would introduce the genetic diversity, and then through vegetative propagation, which could then be used to bulk up the individuals. And so we can then use the information we found about the genotypes and which clones, where the clones are situated, to allow us to perform targeted searches that we could target specific genotypes, perform specific pollinations, and protect. This is the student Kaylee doing a pollination, very careful thing that you do with a paintbrush in the field, but doing targeted pollination events and protecting those pollinated flowers in the hope that you get them through to maturity. The second point here was to protect, and I think here we need to sort of expand the notion of protecting or preventing bark harvesting. That's not the only form of protection we need to think about. We also need to think about protecting genetic diversity. And of course, these things are always limited by resources in terms of manpower and such. And so the one way to approach this would be to think about proportional resource allocation. Do we push more resources towards those populations that contain the highest genetic contribution, those populations which have unique genes. That would be one way of doing it. The other way is to saturate the market by, continuing with the multiply program to sort of reduce pressure on the wild population, getting more trees into ex situ gene bank programs, and even considering the possibility of rewilding, introducing trees into areas where they used to occur but where they have become locally extinct. The third pillar was to monitor. Okay, obviously the first step here from a Kruger point of view is to reassess the Kruger population. We need to establish the effective population based on the genotypes, not on the number of stems. Okay, and so because of that, we need to take these findings from the earlier surveys and we kind of throw them out the window. Okay, these size class distributions don't mean anything in a population that is persisting through the production of clones. It would be really useful to compare the Kruger population to other populations to try and inform us as to why the Kruger population seems to be doing so badly. In terms of monitoring, we need to think, if we go back to those life cycles I presented at the beginning, and we need to think beyond just the standing stems. We need to think about all the pre-emergent parts of the life cycle as well: seeds, the fruit, the germination, the recruitment, the pollination, all those things. I think the monitoring needs to be holistic. It needs to be long-term. And I focus on the long-term here because whatever intervention we implement, there's going to be a lag effect between what we do and how the population responds. Okay, so there's going to be needing to be some monitoring around that. And then the last point here, which I've added in, is I think we also need to have a change in mindset. Okay, if Warburgia was a charismatic animal, like a cheetah or a rhino, and if we were reporting the failed reproductive rates that we are reporting, and if we were reporting the low population sizes that we are reporting, the species would be getting a lot more attention than what it's currently getting. I also do think we need to acknowledge that in terms of the Kruger population, there is going to be a defined endpoint. We need to think about the species beyond just being a medicinal resource. There are other reasons why the species is important. And then we need to go beyond, and I think this sort of my involvement in this program has really demonstrated that in order to understand what's going on here, you can't just do monitoring, you need to prioritize research as well. And one example of that, which we've just started, is some climate niche modeling for the species to try and inform us, sort of to really get at that hypothesis too about the disconnect between the current distribution of the species and sort of the mismatch between where it occurs and the environmental conditions where it's sitting at the moment. Yeah, so I do need to just sort of thank or acknowledge at least a lot of people. There's two fantastic guides, or sorry, rangers rather, Simon and Venus, who know these plants absolutely intimately. They patrol these populations. They were critical in guiding us to trees that had flowers, guiding us to trees that had fruits. Obviously, there's a whole bunch of sort of admin and sort of logistics people in Kruger that supported the population. These guys as well, yeah. Some field assistants, some people from other organizations like the Research Council and Endangered Wildlife Trust, who also have programs running with the species, and then just a bunch of entomologists who help us with some of the species IDs. Oh, so that's my story. Thanks for listening.
Thanks, Dave. Thanks. Great to see a study that spans all the way from genes to population dynamics. Okay, let's see. I think we've got a question or two in the chat. It's a question from Tumbedzo. Tumbedzo, are you online? Do you want to ask your question out loud?
Yes, Tony, I'm online.
Oh, yeah, please go ahead.
Okay, nice talk, Dave. I just want to know what will be the consequences of losing the Kruger Park populations, given that they are genetically different from those that are occurring outside the park.
Thanks, Tumbedzo. I mean, I think so at the population level, in terms of at the South African scale, you want to be mimicked, or the situation is mimicked outside the park, as it is inside the park. So you want to be maintaining as much genetic diversity as you can across the entire population. So losing the Kruger gene pool has implications for the South African gene pool, and likely knock-on effects for the persistence of the species in South Africa.
Okay, thanks.
And sorry, just if I can just add on there as well, and I don't know if I mentioned it in the talk, but the Kruger population is probably one of the biggest remaining populations in South Africa. So from that point of view, it's also critical that it can be, the genes can be preserved if possible.
Bruce, are you there? I see you've also got a question.
Yeah, thanks, Tony.
Yeah.
I'm not going to come online because I've got a bad connection. Can you hear me all right?
Yeah, we can hear you.
Dave, the Kruger population from the distribution map you had up right in the beginning seemed to be right on the western edge of the distribution. And I know that with rainfall and temperature shifts, that area has undergone quite significant rainfall and temperature shifts. Is it likely that this is just a response to changing climate? And what about the Maputo population? Because that seems to be subject to a different climatic regime. Are we seeing the same sort of thing in Maputo, just off the top of your head?
Yeah, so thanks, Bruce. So yes, the Kruger population is interesting because it occurs in sort of climatic habitat, which is quite different from elsewhere in its distribution. It's quite a bit drier and quite a bit hotter than elsewhere. And so yes, it could be that some threshold has been passed where it is now too hot or too dry, or both, for the plants to have the capacity to survive. And I think that that Eastern population, that artificial population, sort of supports that, in that given sufficient rainfall, that the species is actually very productive. We have not looked at Maputo land, but from what I understand, that the species does have quite low sexual output there as well. But like I said, the comparison of Kruger to other populations is going to be key in trying to unpack this further.
Okay, thanks. And I saw another question. Kathleen, are you with us?
Yes, I'm here.
Yeah.
Thanks, Tony.
Thanks.
Thanks, Dave. That talk was incredibly comprehensive and absolutely fascinating. I am intrigued by what you mean by resources, and this idea that perhaps, you mentioned these joined loci, that there might be a genetic failure in the plants to translocate, desirable elements at certain reproductive stages, and looking at, the different fruits of the aborted or viable, and how the stoichiometric ratios vary, and relate that to the environment, and perhaps address this question of what are the genes failing to do. Is that something you're going to explore, or is it too vague?
Yeah, at this stage, we have not sort of tried to link the two hypotheses together. We've got sort of the possible genetic failure on the one side, and then on the other side, the resource limitation, which is really just that the plants are too stressed, either heat stressed or water limited, and that they sort of shift their focus towards just maintaining growth rather than investing in reproduction, and that's very common in plants. At this stage, I had not thought to try and link the two. I certainly don't have the genetic molecular background to do that, but if there are people who can do it, I think that would be really useful.
Just to carry on that comment, sorry to take up the floor, it might be worthwhile just sending a few samples to look at what the carbon, nitrogen, phosphorus, ... ratios are, and see if, you know, what's in the environment, what's in the plants, and
Right.
perhaps the blank space that's left might be then guide you towards some more genetic work.
Right. And I think, you know, now that we've been able to isolate the different clones, and we can pinpoint which clones did have the reproductive output, we then can do these sort of targeted selections of tissue and send off, you know, material from plants that do reproduce, material from plants that don't reproduce, and like you say, compare their resource allocations. Yeah, thanks, guys.
Okay. Yeah, thanks. And I see we've got a hand up from Michelle Hofmeyer. Michelle's not part of SAEON, but she was very much involved in this project in the beginning. Michelle, are you there?
She was there.
Can you hear me?
Yeah.
I'm sorry.
Yes.
Hi, everybody. Thanks for the opportunity. I'm coming to you from Mtinzini and KZN, and the signal is very poor. Thanks, Dave. Fantastic presentation. Really, such a comprehensive update of the work that you guys have been doing. Yeah, it's really excellent. Just 2 points which might be useful for you to note, Dave. There is a gene bank. There's actually 2 gene banks of ex situ population of the Kruger population. One at Sandby, at the Botanical Gardens, Lothar Botanical Gardens, and it's probably like 7 or 8 years old now. So those trees will be getting close to producing fruit, and it'll be interesting to see if any of these hypotheses apply to the ex situ populations. And Karen Hanberg, who's also on the call here now, they've set up an orchard of ex situ plants at ARC in Nelspruit. So there might be an opportunity for you to link up there and see how they're progressing. And then just a comment on the Phinda population, which I assume Bruce is referring to as the Maputo-land population. I'm not sure if that's what Bruce is referring to. We've surveyed those plants together with and beyond, and their reproductive output is astounding.
You're right.
It's incredible. It's the same level as the Eastern plants, even with the Salutaris, even with the fruit fly. I can share that data with you if you're interested. And they actually have kickstarted an entire distribution project based on what the learning was from Kruger. So yeah, we've had some stochastic results with the plants, but in that Sandveld area of Phinda, there's about 1,200 plants, and then the groves are massive. So you could probably extend the study to there to look at your clonal issues as well. So yeah, well done. I think you've set the bar very high. So we look forward to, you know, and we're happy to collaborate if you guys are interested. Thank you.
Oh, great. Thanks, Michelle. I think that's really useful. And you know, the first step there might be just getting some material from those Phinda populations just to look at the genetic diversity there as well, just as kind of a baseline for further comparison.
Absolutely. Yeah, fantastic. And they've even had their first community distribution. So the plants they're being planted back into the local communities, which is a real win for the conservation of the plants, because they're from known location, from a protected area. And the plants in Phinda are also subject to harvesting, even though they are within a protected area. So they have the same constraints that Kruger has, although probably, and maybe just one comment, if I may, on the plants in Kruger. And I'm sure you guys agree, they're at the end of the distribution on the Zoutpansberg. So if you step back and look at what the populations look like closer with more stable rainfall, you'll probably find, if you had to collate weather data, it probably would support your resource stress with the stress response to higher temperatures, the frequent droughts that we've had. So maybe you want to extrapolate some of the data from the long term, because I know Tony and them were crawling around in those Warburgias many years ago and collecting the data. So there is a reasonable data set on that. So yeah, thanks very much. You guys are doing incredible work. Really fantastic.
Great.
Thank you.
Thanks, Michelle. Good to hear from you.
Okay, thanks. I don't see any more questions coming up. And we're pretty much filled up our hour, so I think maybe we should wrap it up. So thanks again, Dave, for that interesting talk. And thanks for everyone who attended and for your continued support for the series. And remember, it will be up on YouTube as well. And then just to end off, a reminder that next month we continue. And it's the grasslands forest wetlands nodes turn next month. And Sue van Rensburg is going to be speaking. And the title of that is Wicked Problems in the Maputoland Coastal Plain, a case for using social ecological systems approaches and the need for transdisciplinary integration. And that is scheduled for 7th October. All right, so please remember that one. And thanks, everyone, and have a good weekend.
Great. Thanks, Tony.
Thanks, sir.
Thanks. Bye.